Okay, and we are on. So everybody, welcome to this new installment of the International Legume Society webinars. For this December session, we have a very special event. For the first time, we are opening our event to young researchers, and today we have three of them: Giudice, Escudero, Feliu, Gaetano, Giudice, and Rafael Duarte. And so we are going to split our usual hour in three, with about 15-minute presentation and a few minutes for question for each one of them. Since we are risking going late, as it happens when there are many moving parts, so let's start right away with Giudice Escudero Feliu. She's a postdoctoral researcher at the Spanish National Research Council, the CSIC, and university professor at the European University of Valencia, Department of Basic Science. So, Julia, please, the mic is yours.
Okay, thank you very much. So good morning, everybody. So thank you very much, Nelson, for your presentation. Today, I will be talking about a part of my PhD research and also a part of my postdoctoral and current research that is about exploring lupin-derived nutraceutical proteins as novel preventive and therapeutic strategies against inflammatory-related diseases. Concretely, I am going to speak about breast cancer. So, sorry, okay. So breast cancer is a type of malignant tumor that is beginning in the breast tissue and arises from the transformation of normal cells into tumors as a result of the interaction between genetic factors and also external agents. As you may see in the image, is the most common tumor in women and also is the fifth most deadly tumor worldwide. So despite all of the technical and medical advances, we still experience a lot of cancer drug resistance to treatments. This resistance is partially and in a lot of times explained by the existence of a subpopulation that is called cancer stem cells that has quite a few interesting properties in terms of metastatic and resistant treatment against the current drugs that we have in the market. So only these type of cells are capable of forming new tumors once one of them is already killed or already treated by any treatment. So cancer stem cells have an increased tumorigenic potential and also have an unlimited capacity of cell renewal. They can also differentiate in any cell that they want. This is why they are so malignant in terms of relapse of tumors after 5 years, for example, of treatment. So we have studied that we have a lot more tumor relapses after conventional cancer therapies, and after cancer stem cell specific therapies, the tumor regression appears because of this killing of this specific population. So apart from that, it's also important to talk about the risk factor and the importance of nutrition in breast cancer. So numerous studies have described the protective effect of a healthy diet in reducing the risk of breast cancer worldwide, and also the relationship between the consumption of some natural products with a higher survival rate in this case. So in this sense, a strong interest is focused on legumes, and particularly we are going to speak today about lupins. Okay, so as you may know, lupin is a legume of the Fabaceae family, and in particular, the seeds of Lupinus angustifolius, or known as blue lupin, is the one that we are going to study in this case. So the main compound of these seeds, it's a conglutine protein that is called beta-conglutine protein, and they belong to the vicillin family of proteins and have been characterized by cDNA clones derived from developing seeds, identifying 7 functional genes that appear on screen, and also 7 functional proteins called B1 to beta 7. So regarding their molecular and nutraceutical properties, our group has done a lot of studies, as you can see in picture, in terms of trying to decipherate what are the molecular aspects of these proteins that could be beneficial for human health. And they find out that we have beta-conglutine multifunctionality. Particularly in a study conducted by my PhD supervisor, Jose Carlos Jimenez Lopez, in 2022, they found out that there were a lot of molecular nutraceutical properties of these narrow-lived lupin beta-conglutine proteins in inflammatory diseases like, for example, type 2 diabetes. Concretely, we can see on screen that we have anti-inflammatory, anti-diabetic potential, also the regulation of pro-inflammatory mediator, also increasing capacity and reducing chemotactic capacity of cells. So due to all of these properties that have already been discovered by previous studies, we thought that beta-1, beta-3, and beta-6, that were the ones that exerted these anti-inflammatory properties, could be used as a strategy for cardiovascular diseases, type 2 diabetes, obesity, and among others. It is also quite important to talk about chronic inflammation as one of the main roots and main factors that can support and help cancer growth. So chronic inflammation is fundamental and is one of the fundamental causes of many diseases and can help to spread the cancer. So because of that and because of all that I told you in the introduction, we thought that maybe beta-conglutine proteins could be an interesting strategy against breast cancer. So the hypothesis of my PhD and the one that we are still currently work on, but in a more advanced level, was that those proteins from blue lupin may act as natural cytotoxic agents against these breast cancer cell lines due to this anti-inflammatory strong effect, also anti-diabetic one. And we also thought that it could have a radiosensitizing effect mediated by the involvement of some important genes and also mediated by the fact that they could potentially affect this cancer stem cell population in which we talk about in the introduction. So for the aim of discovering that, we had two main objectives. The first one was to obtain and purify these three previously studied isoform of beta-conglutine from recombinant ways in order to be able to produce them in larger quantities and use them as a treatment in this type of cancers. So we checked their efficacy as a simple treatment in an in vitro model that I will explain now. And also as a second objective, once we found out their effects in an in vitro model, we wanted to evaluate the potential of them being a nutraceutical complementary technique in order to improve the radiosensitizing potential of conventional techniques in breast cancer. So in regarding for the first objective that we have, we did the conglutines overexpression using an SDA page analysis, and we found out these three isoforms of beta-1, beta-3, and beta-6 in a very high percentage of purity, so we could be able to use them as a treatment in our in vitro cells. So first, we started with an in vitro model in which we wanted to simulate like the reality of breast cancer in our population, that is that it's a very heterogeneous disease. So we started with a healthy non-tumorigenic epithelial cell that was simulating the normal breast cancer cells. And then we studied three different type of cancer cells, as you can see in the picture, that are from best to worst prognosis, okay? Being the triple negative one, the one that is more resistant to any treatment, and the one that has worse prognosis in the real medical world. So first of all, we wanted to see what was the simple effect of these beta-conglutines extracted from blue lupin, and in this case from recombinant ways. And we assessed two techniques of evaluating this type of viability. So our results, as we can see in pictures, showed that you can see in red, we have the healthy cells, and in the rest of colors, we have the tumoral cells. So specifically, we can see that beta-1 was the one that was providing more effect. And we can see that it's we can see very clearly that even if at high doses that are around 10 nanograms per microliter was considered a high dose, we can see some affection from the healthy lines. In the most small concentration, we could see we couldn't see any effect on the healthy cell lines, but we could see a really significant effect in terms of cell growth in vitro for the rest of the cell lines. So we wanted to study more in detail this effect. So we performed an apoptosis assay, and we found out that there was no apoptosis induced in the healthy cell lines that we can see as the first graph. And we see a lot of apoptosis reaching up until 40% of apoptosis in the cancerous cells. So we also wanted to assess if this type of apoptosis was caspase dependent or not. And we could see by a Western blot analysis that we didn't see any activation of the cleaved caspase 3 or either caspase 3. So this means that the apoptosis that we can have in our cancer cells due to this lupin-derived product is going to be caspase independent. Because normally breast cancer cells have defects on the caspase signaling that can resist to conventional treatments that normally induce this type of apoptosis. So this treatment could also be like a new perspective in a way of inducing apoptosis in a different manner in order for decreasing the resistance to conventional treatments. Also, we wanted to study two mechanisms that are really, really important in the terms of biological effect of the cancer cells, that are the ROS expression and also the DNA damage assays. So as you can see in the picture, we found out that with the treatment of our 3 beta-conglutins, we could observe that there was a reduction of the ROS levels and also the DNA damage levels in all of our cells compared to the normal healthy cells. Okay, so a lot of students have already talked about the fact that modulating ROS could be a strategy to overcome multi-drug resistance in cancer, and also that some therapies that were specifically regulating this mitochondrial oxidative phosphorylation could be very useful in cancer. So apart from that, we know that the activation of ROS could cause DNA mutation. So ROS and DNA damage are really, really related to metastasis and relapse in a lot of cancer cells. So because of that, this treatment could not only be an effective treatment in terms of killing the cells, but also in terms of preventing new DNA mutation because of this augmentation of ROS levels that could potentially lead to metastasis and relapse after a few years. Finally, because I've talked about this population in the introduction that was so reactive to the treatments, we also wanted to assess what happened with this more complicated population after the treatment with the beta-conglutins. And we found out lower levels of this population in terms of quantification and also characterization. We performed an ALDH1 plus study in which we could see that we had a diminution of these markers after the treatment of beta-conglutins, which is a positive sign because normally cancer stem cells have higher levels of these markers. And the same with the CD44 and CD24 population in all of the cells. We could see a really dramatic reduction of the phenotype of this population. We also assessed this auto or self-renewal capacity in this model with a mammosphere assay that consists of studying how the cells can, after treatment, renew their capacities and form colonies. And we also confirmed the results that the beta-conglutins could regulate the stemness phenotype in the breast cancer in vitro. We could see that this effect is only appearing in SKBR3 and MDA 2031 cells that are the most aggressive and also have a P53 state of the cell that is different from the normal ones. So we could potentially think that this effect would be regulated by P53. Finally, all of the results were published in 2023. This was the paper of my PhD, in which we described all of these functioning. So we were able to explain that in healthy epithelial cells, the effect of these beta-conglutins is not really important and hasn't got any affection at the concentration that we use in terms of survival. The cells were surviving and were healthy as they were, but it had a lot of positive effects in terms of beating cancer in the different breast cancer cell lines. We could talk about a regulation of some autophagic and some protein that are really important in the development of cancer, and also a cancer stem cell regulation that could be a really preventive strategy for not having relapses after a few years. So as a second objective, we also wanted to try to think about something maybe more close to a market or to a potential patent distribution after our research. So we thought that maybe because we had already a lot of treatments that could have a cytotoxic effect on cells, it would be interesting to try to improve the already existing treatments like radiation. So we wanted to try if the combination of radiation and the effect of this beta-congluten could have a synergistic effect, not only an added effect, and maybe we were able to propose a combination of these two treatments so we could reduce radiotherapy session that also can have some damaging effects in terms of new DNA mutation and a lot of secondary effects in women that have to suffer a lot of radiation processes. So we repeated the same experiment that I have already explained to you with a combination of these beta-congluten treatments, and after that we irradiated in order to see if these beta-conglutens could maybe prepare or help the tumor cells in order to be more sensitive to radiation. So in a clinical term, we could reduce the number of sessions of these radiation techniques. So we did the first colony assay in which we were able to see that in the healthy cells, so MCF10A, we have no effect of the combination of beta-conglutens with radiation, meaning that the healthy cell still are really resistant to radiation in all of the combinations. So when they are alone or when they are treated with our beta-conglutens. But we can see that in MCF7 and the other two more malignant cell lines, we could see that we could obtain the same results obtained normally with 6 or 8 grays, that is quite a really high concentration for radiation therapy. We could obtain them really with 2 or 4 grays with a combination of both techniques. We have already seen in bibliography that we have some radiosensitizer potential of other compounds, for example, resveratrol that is present in peanuts. So this is a really important strategy in terms of trying to reduce the number of sessions of radiotherapy in patients after the consumption of some proteins, like for example, these beta-conglutens of some other compounds are resveratrol. We also wanted to assess apoptosis, and we found out again that there was a really high increased apoptosis when we combined those treatments compared with only the radiation treatment. And also we wanted to quantify again this cancer stem cell population, because one of the failures of radiotherapy treatment is specifically due to the fact that a lot of times radiotherapy itself is unable to kill this specific population. So we wanted again to assess what was the effect of this protein derived from blue lupin in this treatment. So again, we found out that we had lower level and lower doses of both ALDH1 positive and CD44 high, CD24 low markers, meaning that the combination of these two techniques was a really effective way of reducing the CSC population. On top of that, we also performed this mammosphere assay in order to see the self-renewal capacity of cells after the combined treatment, and we saw that beta-conglutens could reduce this phenotype and self-renewal capacity in all cell lines at any condition. So the conclusion of our of my PhD project, and also some of the newest approaches that we have now, was that first we have a broad therapeutic potential of congluten proteins in the market. So congluten proteins are shown a wide therapeutic potential against breast cancer. They could be used as natural cytotoxic agents and also key modulators of pathways that are involved in tumor cell survival, treatment resistance, and also metastasis. Finally, they can also impact on this cancer stem cell population and self-renewal. So this could be also a contribution to preventing tumor progression, metastasis, and recurrence. So we can have maybe nutraceutical added to the natural or normal treatment of breast cancer patients in order to help preventing the tumor progression. Finally, the synergy with radiotherapy also opens the door for future therapeutic application, being a promising avenue for example, combined therapeutic strategies, and also suggesting a potential use in prevention, advanced stage treatment, and also reducing the relapse risk. Also, I wanted to talk about the next steps that we are currently doing. So we wanted, the next step of our research is to prove the efficacy of this model in an in vivo treatment that we are currently doing. Also, we wanted to elucidate exactly what is the mechanism of action of proteins congluten beta, if they are able to enter the cell or they act through some receptors, but we want to elucidate what is the mechanism of action, and we are currently doing assays for that, and also to study what would be the better way of administration, if it could be maybe through nutrition strategies or through administration toxicology assays. So we are also currently studying that. Also, we have a published patent nowadays that you can check in the link that I'm appearing, of the use of these conglutine beta proteins in combination with existing treatments in order to improve the quality of, for example, radiotherapy treatments. We also want to do all of these studies in order to, in the future, be able to propose a clinical trial so we could have more data about this effect and maybe try to do like a natural medicine therapy from proteins that were previously found and present in lupin. Also, we want to do more studies about other nutraceutical compounds in this way and other type of inflammatory diseases like cancer and others too. And finally, I also wanted to talk about the fact that we have a lot of future perspective from this perspective from nutraceutical proteins to legume-derived anti-cancer strategies. So first, it would be really interesting to study natural variability in lupin. So to see if Lupinus angustifolis varieties could have different isoform or function of the beta-conglutine proteins, not only in a nutritional aspect, but as a treatment, okay? Also, it would be really, really interesting to do genetic improvement tests. So to see, for example, breeding programs that could be implemented to enhance, for example, the total protein content, so have more protein content of this protein in the lupin, the proportion of conglutine with greater bioactive potential, or the reduction of anti-nutritional compounds. Also, we can talk about food processing effects or functional extract and nutraceutical ingredients that could be the first step of developing lupin-based nutraceutical products that could have application in prevention, adjuvant therapy, or just supportive care for patients that could be at risk of having some of these diseases. Finally, it's important to talk about the expansion to other legumes because if we have been able to find some compounds in lupin that are really good anti-cancer strategies, I have already talked about the fact that found in some of them also in peanuts. So I'm sure that we could potentially found other anti-cancer strategies there. And finally, before finishing, I wanted to thank all of the institutions that have supported my research during the years.
Yeah.
Also thank the International Legume Society for inviting me to this event. And I wanted to say that this multidisciplinary approach, I think, aligns perfectly with the mission of the International Legume Society, that is strengthening the bridge between crop science, human health, and also molecular nutrition. So conglutines could represent a promising example of how legumes contribute not only to sustainable food, but also to innovative biomedical applications. So thank you very much for your attention, and I'm happy to answer the questions that you may have.
Thanks, Julia. It was a very interesting contribution. It's also new. It's a new kind of topic for us. We just brushed the nutraceutical effect in a previous presentation. There are a few questions from the chat, but you almost already answered to all of them because the questions were asked before your final slides. So somebody was asking about other varieties of lupin, Lupinus albus, etc., or fava bean, other legumes, because our base is still of breeders and
Yes.
and a lot of people working with other aims in mind, but you touched on them. And just a quick answer from your side. There is somebody asking, what about the difference between consumption of whole lupin seeds versus just the beta-conglutines or the specific proteins? Is something that it's on the horizon?
Okay, so there are, I think there are published a lot of, well, not a lot, but quite a few studies that work with the whole group of proteins from breeding or from the legume seeds, and also found out that there was an anti-cancer effect. But it's true that the fact of working precisely with each one of the proteins or each one of the isoform gave us much more information about their different properties. And also we are able to know that it's only because of the effect of this protein and not because maybe of the combination of them. Also, we think that in terms of producing, for example, a potential new treatment, it would be easier to concretely produce only the sequences that are more interesting because, for example, in previous studies that our group had, when we use the combination of all of the conglutins or just the processed protein from the seed, the results were lower. Okay, so that means that in terms of nutrition, it's interesting for people to know that legumes, and specifically lupin, are really, really good in terms of concentration of proteins, and the proteins are really helpful for our health. But if we want to focus it more on bioactive compounds that could be a medical treatment, we think that is more appropriate to produce them through recombinant ways and only produce the structures that are important. It would also be really important to talk maybe potentially about allergies and how to prevent them if patients could be maybe allergic to some of the compounds.
Okay, thanks, Julia. Thanks for the answer. Okay,
thank you very much.
So since as is tradition, we are running a little bit late, we move to the next presenter, which will be Gaetano Giudice. So, Gaetano, please share your screen. Gaetano is a postdoctoral fellow at the Department of Soil, Plant and Food Science at the University of Bari, AldoMoro, here in Italy. And when you're ready, you can start, Gaetano.
Oh, thank you, Nelson, for your presentation. Good morning, everyone. I want to thank the International Legume Society for giving me the opportunity to present our research about resistance mechanism to Orobanche crenata in pig. So, Orobanche crenata belongs to the family of Orobanchaceae, and this is also known as broom rape. This family groups about 2,000 species of parasitic plants, and Orobanche crenata is widespread in the Mediterranean basin, with devastating effects on the cultivation of good seeds of legume, determining up to complete yield loss in pig. And in these pictures, we can see the effect of the plant parasitization that leads to complete death of the plant. So Orobanche crenata life cycle, like other species belonging to the family of Orobanchaceae, starts with the seed germination. Then we have the formation of soia, that is the feeding organ of the parasites, and the attachment to the root host. Then it proceeds with the tuberculous formations, emergence of the flowering shoots, flowering, fruiting, and seed dispersal. I would like to point out that Orobanche crenata seeds need to fill strigolactones, that are carotenoid-derived compound, to germinate. These compounds are produced by the host plants and secreted in the rhizosphere, and are perceived as germination signals by the broom rape seeds. So far, chemical and agronomic approaches for the control of Orobanche crenata have shown limited effects, and breeding for resistance is of major importance and is considered the most economically and environmentally friendly control method for the broom rape management. So, resistance mechanisms could take place at several stages of the Orobanche crenata life cycles. We have resistance mechanisms that acts at the pre-attachment stage, where plants produce a low level of strigolactones or other kind of germination stimulants, or an altered composition of molecules. Resistance mechanisms that acts at the pre-ostoral stage, as we can see in the pictures on the left, usually involve the deployment of physical barriers or the accumulation of toxic compounds. Finally, resistance mechanisms that act at the post-ostoral stage usually involve necrosis and death of broom rape nodules or the sealing of the host vessel. So a few years ago, my research group reported the selection of nearly complete resistance pig bedding line that is named the ROR12. And in these pictures, we can see how it behaves compared to the susceptible cultivar sprinter. And it's possible to note at the base of the sprinter plant several shoots of the parasites, while none are present under ROR12. That looks also much healthier. So the characterization of the root exudates showed that ROR12 produces significant low strigolactone level compared to the other cultivars for all the three strigolactones that are produced in P, orobanchol acetate, and fabacil acetate. Moreover, used a QTL mapping approach in order to identify particularly QTLs that are associated with ROR12 resistance. And we used a population of recombinant in red lines with 147 individuals that were obtained by crossing ROR12 for the susceptible cultivar sprinter. This led to the identification of three QTL, PS-OCR1, PS-OCR2, and PS-OCR3. So the aim of the study is the investigation of the role of P-strigolactone in the field resistance to Orobanche crenata. To fill this aim, we used three different kinds of plant materials: a recombinant in red line population, a global P-gem plant collection, and P-strigolactone by synthetic mutants. Concerning the recombinant implant line, we selected 8 lines carrying all the possible homozygous allelic combination at the 3 quantitative trait loci. So for each loci, we had 4 lines that had the resistance allele in homozygous configuration, and 4 lines carrying the susceptible allele in homozygous configuration, and all the possible combination. Then we proceeded in collaboration with the University of Amsterdam to quantify the strigolactone from the root exudates. So we compared the average strigolactone levels between the recombinant implant line that were homozygous for the susceptible and the resistance allele. And as you can see from the table, only PS-OCR1 was significantly associated with a lower strigolactone content, differently from PS-OCR2 and PS-OCR3. And this suggests that ROR12 have different resistance mechanisms that are strigolactone dependent and strigolactone independent. In addition, to further characterize the role of strigolactone in the field resistance to Orobanche crenata, we also used a P-gem collection of 250 pure lines. We performed field trials to observe the response to crenate brome rate, and also quantified the strigolactones in the root exudates in 6 P lines that were selected for contrasting field response to crenate brome rate. As you can see in the graphic on the left, we selected the most 3 resistant line and the most 3 susceptible lines. Then we analyzed the strigolactones in the root exudates, and the resistant line had a significantly lower strigolactones content in the exudates, and these lead us to the conclusion that the resistance is associated with a reduced strigolactone exudation. Then we wondered what could be the level of parasitization for plants that do not produce strigolactones at all. So we used 2 mutants, the RMS1 mutants and the RMS5 mutants, that were impaired in the production of strigolactones, since they were defective for 2 enzymes in the biosynthetic pathway of the strigolactones, CCD7 and CCD8, and then proceeded to quantify the strigolactone from the root exudates, and also performed a field trial to observe the response to crenate brome rate. As expected, no strigolactones were detected in the mutants compared to the wild type release. Looking at the results of the field trials, here as well, it was not surprising that the 2 mutants had a lower level of parasitization compared to the wild type release. However, interestingly, they had a higher level of parasitization compared to the ROR12 line, which showed a almost complete resistance to the parasite. So this led us to conclude that strigolactone deficiency alone is not sufficient to confer complete field resistance against Orobanche crenata in pig. To conclude, lower strigolactone production is an important mechanism for the resistance to room rape. However, it does not provide a full resistance, at least in pig. The complete absence of strigolactone can also have negative pleiotropic effects on the plant, as we can see in the plant on the right, which shows extreme branching. So, complete and durable field resistance can be achieved by combining low strigolactone production with other mechanisms, as in the case of ROR12. As switch perspectives, we want to proceed with the identification and functional characterization of candidate genes in the QTL regions and perform genome-wide association studies to identify marker phenotype association signals. Finally, I want to thank Professor Stefano Paman, who coordinated the research, and all our collaborators from the University of Foggia, the University of Amsterdam, the CNR UBR Institute, and the INRE. And thank you for your attention. And I'm welcome to answer your questions.
Thanks, Gaetano. Your presentation was very on spot and very interesting, given the interest in Orobanche, which I guess it's also a growing concern in European agriculture. Are there any questions from the chat or from the group? Okay, so Tom is asking what I was going to ask too. So apart from strigolactone, what would be do you have an idea what would be the source of resistance or the resistant mechanism in ROR?
So in ROR12, we are sure that some of the resistance is given by reducing strigolactone production and secretion at the rhizosphere. However, it's one of our aim to perform future studies in order to look at those genes that are in the interval of the QTL that was not associated with the reduced production of strigolactones. So this is one approach that we want to use.
Okay, so good luck with that and with this investigation. So if there's no more questions from the chat, I'll thank again Gaetano, and we move to the third and last speaker for today, Rafael Duarte, which is a PhD student in biotechnology and research at CBQF, the Catholic University of Portugal. So I guess that Gaetano, you should stop sharing your screen. Thanks.
And, hello everyone, thanks. I'm just going to now share my screen. Perfect. Thank you, Nelson, for the presentation.
Sure.
So hopefully it should be working well. Let me just full screen.
You should go full screen.
Okay. So hello everyone. Thank you, Nelson, and thank you all for joining this session. Today I will be talking about why legumes are much more than just protein sources, and I will explore how their non-nutritional phytochemicals influence both soil and gut microbiomes, and how this interconnects with the One Health framework. So this will be very quick. My name is Rafael Duarte, and my work sits at the interface between agricultural sustainability, plant biochemistry, and its microbiome science. I did a bachelor's in biochemistry, then a master's in applied microbiology, and I'm now doing a PhD in biotechnology. So, speaking now about the work, One Health is basically the idea that the well-being of humans, animals, plants, and the environment are deeply connected. And for agriculture, this means that we must rethink farming, not only in terms of yield and protein, but also in how it shapes soil ecology, which in turn influences our food system and ultimately our own gut microbiomes. This is crucial for the entire ecosystem health. And I am sure a lot of you know this, but what do we call anti-nutritionals or non-nutritionals are basically phytochemicals, and we have a huge variety of phytochemicals since very small to large molecules. And as you can see here, we have a simple phenolic acid or lignin, which is a very complex compound. And normally these compounds are considered as anti-nutrients. They are being perceived as bad for both humans and animals, but they protect plants, and they are only produced by plants for their protection. They are secondary metabolites, and some of the main classes include phenolic compounds, alkaloids, terpenoids, and others. And also, sorry, they have strong antioxidant capacities. But historically, as I was also highlighted this, legumes were being bred to reduce compounds like polyphenols or lectins, but new evidence shows that these so-called anti-nutrients can have a positive benefit, and and more than a positive benefit, they can have a dual benefit both for gut and soil. So why not in future breeding we are optimizing these compounds more than just breathing out these compounds. And for this study, for my PhD study, I have chosen legumes, and I will not expand much here because most of you knows of the benefits that legumes have. But basically, uh, our focus here, it was since we are aiming for a two-pronged approach, both human health and environment health. Legumes were an easy choice, and within legumes we have chosen to work with pea and fava or field bean, as you would rather. And in this study, we were also involved in these two parallel approaches that we started with the chemical analysis of the phenolics and of the phytochemicals of our material, either grain or the plant or the other parts of the plant. So we had to know what we had, and then we had to test like these different phenolic compounds and these different legumes in regards with the soil and gut ecology. And even though this is different, what we do in the soil is not the same to what we do in the gut. Both systems, we end up studying the microbial diversity in the soil microbiome and in gut microbiome. We end up seeing how these contrast responsive genotypes, higher phenolic or low phenolic, impacted this diversity, and how it impacted the nutrients involved in these systems. In the case of the gut, the short chain fatty acids that I will speak further, and in case of soil, like nitrogen cycling and carbon cycling and others. And because I can start talking about both together, like soil and gut, I will start with soil. And regarding soil, I think you will know that agriculture needs organic inputs. And although a lot of people use like chemical fertilizers, NPK, we can use residues. Meaning what? We know that legumes, while growing in the soil, they are doing good because they are fixating nitrogen from there. But after the legumes are grown and the grain is collected, we have the plants there, and we can use those plants as residues that we put back into the soil, and they are rich in carbon and in nitrogen, and they can also have potentially bioactive compounds that can help this process. And a lot of studies look into the carbon and nitrogen ratio, and there is a reason for that. It's a good way of having an idea how the residues will behave. But in this study, we decided to look further, not only about the carbon and nitrogen ratio, but what these compounds have in regards with their phytochemical profile. And that is how our residue experiment comes in. And in here, in this residue experiment, which was done in Scotland, with help of the James Hutton Institute, we basically took field bean and pea residues of different types, and we incorporated them back into the soil in the mesocosm experiment, and we did that in two incorporation rates, 5 tons per hectare and 20 tons per hectare. not only the dose was varying, it was also varying the phytochemical composition of these residues, because I had a high phenolic and a low phenolic for both P and Faba. And then after putting those residues back into the soil, we grow spring barley. And why spring barley, you might be asking? Because normally when farmers are working in the field, they, after growing legumes, they will grow a cereal. And so it makes sense, like agronomically speaking, to grow barley after a soil that had legumes there. Right? So this is the basic idea, and I was, as I was telling you, in these mesocolumns we use varieties with different phenolic, as we can see here, and different lignin contents. And we don't have time to go through all the data, but as expected, oh, and also we have aimed for similar carbon and nitrogen ratios between them. So the thing that was varying the most, it will be the phytochemical content. And we don't have time to go through all the data, but as expected, higher lignin and higher phenolic genotypes had a composition that was slowed down, which affected nitrogen cycling, microbial responses, and especially plant growth. And we can clearly see that at high doses, residues cause a nitrogen immobilization, meaning what? Microorganisms lock the nitrogen for their own use, reducing plant available nitrogen, and we can clearly see that because of the suppressed barley growth. But at low doses, meaning 5 tons per hectare, which is honestly what most farmers use, either 3 tons per hectare or 5 tons per hectare, so maybe 20 tons per hectare was too much. But at low doses, residues improve the nitrogen availability, and low dose treatments perform best, even comparable to the mineral fertilizer, which has the nitrogen much more available. And this highlights how both residue quality and quantity impacted either the soil parameters and the plant growth. But also, I also have to highlight here, the high phenolic may not have a positive effect in this crop, but there is a potential for future work to see the effects of the high phenolic, high residue into the subsequent crop. Meaning what? Not only after we are growing barley, then we would collect the barley, and then we would grow barley again, because maybe these high phenolic crops and their gradual breakdown may enhance the nitrogen availability in the next cropping cycle. So, and this was one type of our soil studies. We are also doing a legume intercropping, which is kind of new, because normal people use a legume cereal intercropping, but it is an innovative way that legumes can have an impact. And again, phenolic variation played a central role in this design, because we have chosen combinations of high and low Ps and high and low Fabas for the intercropping, and we are comparing them with the monocropping. And we can see here, this is only like preliminary data, but the high phenolic P genotype rows, after being grown in intercropping, even has more phenolic compounds, even has more antioxidant activity, which kind of makes sense, because intercropping is somewhat a more stressful situation for plants than the monocropping, which indeed they will be producing more phenolic compounds. So that is something to have in mind. And we are also currently analyzing the rhizobacterial communities, using both 16S and ITS sequencing. So in a while, we will also be able to check either intercropping, these high phenolic and low phenolic, is having an impact on the gut, on the soil microbiome, either bacteria or fungi. So this is work still currently going on, and hopefully I will have news after. And going now to my work in Portugal, this was work done in partnership with the James Hutton in Scotland and Portugal, but here we have looked about how different pea genotypes behaved in the gut microbiome. And we compared all seeds and hulls. And a lot of you may be thinking, what is the use of using hulls? Because hulls normally have higher phenolic content, and hulls in some industries, they are being sold out as an byproduct. They are being used in animal feed without any value added. but because they have low protein. But they have low protein, yes, but they have more phenolic content. So we decided to look about the effect in the gut of the whole seed and the hulls, because people will not eat hulls. I know that, but there are some people doing studies now of putting like a hull powder composition in like baking powders or like muffins or bread. So that was the reasoning for the gut study. We decided to look about the hulls because they have high phenolic content and they can be used as products. While the whole seed, people will eat the whole seed. And in this study, to assess the impact of these fractions, so first, how are you measuring this? We did a gut fermentation where we simulate the processes that occurs in the human being, and we are measuring short-chain fatty acids. Short-chain fatty acids are metabolites of the gut fermentation, and they are a good way of checking either the fermentation is doing well or not, and also considering the levels of butyrate or propionate or acetate, we can have an idea of what our product, of the effect that our product is having. And obviously, we'll look about gut microbiome. We'll be checking how the different types of either P and FABA are working within our gut. And here we, as you can see, we included several P varieties from commercial to heritage types. We started with all these genotypes, and we then selected the two genotypes, the black badger and the Octavia, which are the high phenolic and low phenolic. And as I told you, we went searching for phytochemicals, not protein. And about the way that we did this, all samples were cooked and digested using the InfoGest model to mimic typical consumption, and then we used them into a controlled fecal fermentations. We included in this experience a negative control and a positive control, the raffinose. Raffinose is a known prebiotic, meaning we know it does well to the gut microbes, and we compare them with the whole seed and the hull of our genotypes. For this part of the study, because it's a lot of stuff, I will speak only about the effect on the peas. And one of the clearest results was the strong production of short-chain fatty acids, especially butyrate, which has a potential to have a prebiotic effect. And both whole seeds and hulls behaved in a similar manner. Obviously, all seeds a little higher, but hulls behaved pretty well. Honestly, behave well than I was expecting, especially on the butyrate production. So, but after doing these the more metabolic parts, we went to look about the microbiome stuff. And what you can see also is that the P treatments shaped the microbial community in ways comparable to the prebiotic, and the communities shifted more over time than honestly through their composition. Meaning what? We don't have a clear cluster for raffinose and our P samples, meaning the P samples are behaving similar to the raffinose. And we can see that here. Here is basically a between group t-test, the difference between our high phenolic, the BBET, the orange one, and the low phenolic, Octavia. And we can see here that we had a higher production of bifidobacterium in the high phenolic, which is normally considered a beneficial prebiotic genus, but an increase in Escherichia shigella, which can be potentially harmful. Meaning what? We also have like a trade-off here, meaning we have to bear in mind that the benefits cannot outweigh the negative effects. But here, since this increase in Escherichia shigella was within levels that remained within the normal range, we can say that the phenolics, the high phenolic genotypes, are doing bad. They are doing good to our gut microbiome. So, in summary, both genotypes and fraction influence the fermentation and the microbiome responses. Peas produce more beneficial metabolites like butyrate and acetate, and together, these results also highlight the potentials of peas to support gut health while encouraging a balanced consideration of the anti-nutritional factors. But in this case, all went well. But a future avenue of doing this work is people will eat the whole seed. Now that we have assessed that the hulls are doing well, perhaps next step is to incorporate these hulls into some baking powder or some other product. And hopefully this work creates a type of soil to plate continuity where we are breathing for beneficial phenolics, using those grains to support nutrition and gut health. And also after we collect the grains, we have the residues, and we can return those residues back to the soil, and therefore supplementing the initial source of this holistic one-way view of doing work. And that's the general idea. I am still finishing my PhD work, so I still have a lot of data to publish. And thank you for your attention, and thank you for both the Scotland partners and the Portuguese partners. And if you have any doubts, please tell me.
Thanks, Rafael. It was a very, very nice, very on-point presentation. So right away, I have something.
Yeah,
if
sure.
you may found positive from the chat, somebody asking for your contact details. I refrain from just typing your email there, but if you want, there is somebody.
Yeah, sure. Let me just check.
Just take a look at the chat, and you can get in contact with.
I will take note of that.
Sure. Are there any questions from the chat or from the audience? Otherwise, so would you like to expand a little bit? So given
I just
also
share my email
sure.
also just for the.
Sure, I see. Thanks. Would you care to expand a little bit, given also our standard audience? So what would be a breeder perspective in your, given what you have found, the contribution of this secondary metabolite? Do you think, and do you see a breeding program that could leverage on that and maybe do some specific breeding for very high phenolic content or that kind of product?
Yeah, like more. We are working with partners either in Scotland and here in Portugal of breeding out new varieties of legumes. Because like these new varieties of legumes, we can combine traits. Like I may be more aiming for the high phenolic content, but if some partner is also aiming for like a genotype that is more resistant to drought or something, we can hopefully like because that, but also that depends a lot on the data. But if we have data, we can normally we can do like we can play both sides. We can choose a genotype that has a higher phenolic content that we are confident that will be good for the gut microbiome or for the soil microbiome in the next crop. But yeah, we are working that, and we are doing more tests. And because people also don't want to lose the protein, farmers are also very keen on the nitrogen content and on the protein. So it will be hard to optimize breeding only for the phenolic compounds, but I also think that's not the desirable. But it's not, we don't have to breed them out.
Okay.
We can combine traits and breeding, start breeding stuff that has high protein content, but also has a higher phenolic content.
Okay, thanks. Nice answer. Do we have any other questions from the group? It appears that no. So once again, I thank all the presenters for today. I think it went really well. And also we are quite sharp on our closing time. So thanks again for everybody. As usual, this is going on our YouTube channel and on our website. And I will see you all next year for the next installation of our webinars. Thanks to everybody. Bye.
Thank you. Bye-bye.